So my project is unraveling the link between oxidative stress and protein aggregation in early Alzheimer's disease formation. So Alzheimer's disease is a progressive neurodegenerative disorder that results in memory loss, cognitive impairment, and ultimately neuronal death or apoptosis. It's the fifth leading cause of death, and just over 7 million people in the United States suffer from Alzheimer's today. So whenever we talk about Alzheimer's, scientists refer to these specifically these amyloid beta plaque formations. Okay, and these plaques are extracellularly formed, and they are quaternary structured proteins. And here, specifically, what we hypothesize that they do is these plaques will sit between two neuron cells and disrupt the cell communication between the two cells. And essentially what this will lead to is neuronal cell apoptosis, which is when two cells cannot communicate with each other. And what happens is they'll die off to their inactivity. Okay, and so over time, in a healthy brain, we see a full-size healthy brain here. In a severe Alzheimer's brain, what happens is it'll slowly start shrinking and shrinking and shrinking, and more cells will slowly start dying off. Usually, whenever somebody is diagnosed with Alzheimer's, it's usually located anywhere between 2 to 10 years from when a patient gets early onset to all the way up to severe dementia or Alzheimer's. Okay, and so where do these amyloid beta plaques form? These amyloid beta plaques actually form through a process called amyloid beta precursor processing. Okay, and we focus on a specific membrane protein called the APP, which is an amyloid beta precursor. And it undergoes normal processing, and one of the byproducts it produces are amyloid beta monomers. Okay, and these monomers will actually go on to form these toxic oligomers. And those oligomers will actually bind to each other and form these long fibrous proteins called fibrils. And those fibrils will form these long, large blob-sized plaques, which is what we see here in this diagram. And as I explained earlier, they will actually block cell communication. So we do have ways to actually detect these amyloid beta plaques through a technology called positron emission tomography. And here we do have a simplified scan here between a normal and Alzheimer's, and there is a clear differentiation between the two. But the implication here is that once we get a positive scan on a patient, essentially it's too late to reverse the formation of these plaques. Okay, we know these as kind of tombstones of the disease. Okay, once it's there, it's not possible to reverse it. Okay, and the second implication we face is that some patients will actually show a positive scan. Okay, but that positive scan on the patient, the patient would be asymptomatic because of that positive scan, which is weird, right? It kind of decreases the reliability of positron emission tomography. Okay, and a lot of scientists are looking into why this actually occurs, right? And a lot of this is actually hand treatment because we can't actually make these plaques just go away. As I said, it's just irreversible. Okay, and so I wanted to actually look more into pre-amyloid formation pathology, essentially what happens before these large plaques form in the brain. Okay, and so the main objective of my project is to see if increasing oxidative stress would actually cause a different protein expression level, specifically protein aggregation. Okay, and if it does, what is the trend? Okay, so as oxidative stress increases, what happens to the plaque formation? Okay, is it linear? Is it exponential? Is it a threshold model? Okay, and originally, my original hypothesis was that yes, there will be a significant difference, and it would actually follow a linear trend. Okay, so moving on to some methodology here. I used yeast has a model cell line. Okay, it is important to note that yeast is not an exact replica of human neuron cells, but they do have conserved pathways, especially linking into metabolics, which is decomposing into oxidative stress. Okay, so the first thing I did was I grew the yeast on a YED agar medium for 24 hours. Okay, and the cells were then picking up and taken into a vial of PBS, which is phosphate buffer saline. That was the medium they sat on while I did this entire experiment, and I got a final concentration of OD 600 equals to 1.2. Essentially, what that means, it's at 600 nanometers of absorbance is 1.2, which correlates to a cell concentration, which I can't think of off the top of my mind. So after the cells were homogenized, I could then aliquot those into different treatment groups. And in this table, you see all the the hydrogen peroxide concentrations I used and the different incubation times, so the two variables. And by the way, the hydrogen peroxide is what I actually use to induce the oxidative stress. Hydrogen peroxide is known to be a reactive oxygen species, or maybe even a reactive oxygen species intermediate that will go off and form superoxide, okay, which is specifically what I'm focusing on here. Okay. Now, after the hydrogen peroxide was actually treated, I could then go off to confirm that if the reactive oxygen species actually formed, which is a superoxide, and I used a fluorescent dye called DCFDA. Essentially, what happens is when DCFDA comes into contact with intracellular ROS, it will go into this reduced form of DCF, and when it's excited at a specific color wave or a color, it'll fluoresce green. Okay, and to detect this, I used a fluorescent microplate reader to detect the fluorescence, actually outputs it into arbitrary units. So looking at this here, as I increase the hydrogen peroxide treatment on my samples, the average fluorescence increased pretty much for all treatments, more or less. There is a slight dip in the 0.4, 0.5. There are variations in there, but for the general trend, as hydrogen peroxide increases, the average fluorescence of DCF does increase in this graph, which does validate my oxidative stress inducement in these cells. Okay, so I could then now go off and quantify what I'm actually interested in, which is the amyloid fibrils. And remember, those fibrils actually go off to form those plaques, which is what causes Alzheimer's, or what is hypothesized to cause Alzheimer's, so we put it that way. And essentially, I went in and stained stained the fibrils using thyroflavin T. Okay, and so how these fibrils have these negative charges on these large fibrous proteins, and thyroflavin T has a nitrogen atom that is positively charged, and essentially what happens is that nitrogen will bind to one of these charges, and when it's excited, it will fluoresce blue. Okay, and then I could now go in and quantify that using a fluorescent microplate reader. Okay, and so I did this to every single treatment group of hydrogen peroxide, including a negative control, which is no hydrogen peroxide treatment, and another negative control with no thyroflavin T treatment, which is the blank. Okay, I use the blank to ensure that the fluorescence is only coming from the thyroflavin T, the actual molecule. Okay, or sorry, the actual protein in this case. And any background noise would actually be subtracted out from the data, and to ensure all the fluorescence is coming from is being emitted from the thyroflavin T being binded to amyloid fibrils. Okay, and so next off, now I had a ton of data. I needed to conduct statistical analysis to see what was the relationship and if there was actually a significant difference between the two. Okay, I conducted two independent t-tests on high treated hydrogen peroxide, which is 1 millimole and 2 millimole in this case. And with the thiouroflavin T incubation time at 4 hours, I saw that at 1 millimolar and 2 millimole, compared to the control, there was a significant difference at 4 hours of incubation time with thiouroflavin T. This graph that you see here is hydrogen peroxide treated at 270 minutes. Essentially what happens is oxidation was removed at the 270 minutes level, which is the highest incubation time treatment. There was more data at 180, 120, 60, 30. It's just not included. Okay. And two, for a thiouroflavin T incubation time of 8 hours, what I noticed is there is a significant difference between a 2 millimole treatment as compared to the control, but there wasn't actually compared to the 1 millimole treatment at a 0.05 alpha level. Okay, and lastly, at a incubation time of 24 hours, so just over 1 whole day, I noticed that there was actually no significant difference at the 0.05 alpha level between 1 and 2 millimole concentration. This is all replicated 8 times, okay, to ensure that there was significant statistical power behind the t-test, okay? And so next, well, let me say this first. So first, there was an actual divergence. There was a kind of a divergence here. Okay, so as we move further and further on with 4, 8, and 28 hours, there becomes less and less thiouroflavin T fluorescence. Okay, and so I wanted to model this on a graph to see if I could find a linear regression, which I originally hypothesized that was going to be linear. But it wasn't actually linear, and I actually had to do some nonlinear regression modeling here. Okay, the 3 different legends that you see here is with hydrogen peroxide treated at 70, 120, and 30 minutes. That's how long it was incubated in the hydrogen peroxide before it was spun down. And the hydrogen peroxide was removed from the cell, and the cell was allowed to sit independently. Okay. So for my nonlinear regression, okay, I tried many mathematical functions, which was linear, exponential, and a logistical function. And the logistical function actually yielded me the lowest sum of squared errors, which actually showed the best model in this. Okay, and so I actually have the equation here in the B section showing this is a logistical function here. It best models it. There is no actual regression here on this graph. In the future, I do want to add it in on this poster. There's no regression on here. So yeah, this trend actually does tell us some very important information. So what we see is a window where hydrogen peroxide concentration is increasing, which is modeling oxidative stress, but the average fluorescence of coming from the fibrils is not. Okay, and once a certain point, which is the inflection point, it would then go off and increase really quickly, but then come back off to essentially a capacity, okay, which is modeled by the logistical function. Now, this does tell us something very important, okay, as increased hydrogen peroxide, there is a level where we can go in with some sort of therapeutic going off of this, based off of this trend. And we could kind of say that oxidative stress is a biomarker or early onset biomarker for Alzheimer's disease, and go in and use specific chelating agents or antioxidants to actually mitigate these things and prevent it from actually reaching that point where it accelerates and these fibrils start to actually form these large plaques. Okay. Yeah. So my conclusion was that as oxidative stress was induced on these yeast cells, okay, it does promote amyloid fibril formation. And as it actually increases, I proved my hypothesis wrong, that this does not actually follow a linear model. It does, but instead it follows a threshold dependent model, where there's a short window, where we can actually go in and start treating these patients, in a specific way, right? Or in a specific using specific agents or antioxidants, right? Yeah. So for future research, I do want to go in and validate these things using Western blotting, which is a specific gel electrophoresis method, um, which once we go undergo gel electrophoresis, we can then stain the antibodies and take a look at we can then stain with antibodies and take a look at the gels under specific imaging, to see if they do actually compare with the actual protein and it's not actually due to other things. I do want to look at the cells under scanning electron microscopy, and take a look at what's actually happening, and particularly maybe the morphology of the cell. Um, but yeah, that is my research.